Good afternoon. I'm very happy to see all of you here. I'm Marie Bernard, co-chair of the Trans NIH Women of Color Committee, which is a subcommittee of the Working Group of Women in Biomedicine. And we were very honored to be able to nominate and find selected Dr. Roberta Diaz Brinton as today's WALS lecturer. Dr. Brinton is the Vanderveen Chair in Therapeutic Discovery and Development at the University of Southern California, where she is Professor of Pharmacology and Pharmaceutical Sciences, Biomedical Engineering, and Neurology. She directs the Norris Foundation Laboratory for Neuroscience Research and the USC STAR Science Education Program. She's internationally recognized as an innovative leader in Alzheimer's research and development of therapeutics to prevent, delay, and treat the disease. Research in her lab focuses on discovery of why the brain can develop Alzheimer's and on translating those discoveries into therapeutics to prevent, delay, and treat the disease. Of particular focus is the aging female brain and elucidating mechanisms that underlie the twofold greater lifetime risk of Alzheimer's disease in women. And thus you can understand why the Women in Biomedical Careers Committee would be interested in having her talk. She has developed therapeutics that restore energy production and neural stem cell regeneration in the brain to prevent and treat Alzheimer's. Outcomes of her discovery and translational research have led to 2 NIH-funded clinical trials. She's been continuously funded by from NIH from her predoctoral years to the present support through my institute, the National Institute on Aging. She has over 180 publications and holds multiple patents for therapeutics targeting Alzheimer's and other neurodegenerative diseases and sustaining neurological health during aging. She communicates the impact of her science to the public through media, where her work has appeared in the New York Times, in over 100 global media outlets, ABC, CBS, Science Friday, and onward. She was awarded the Alzheimer's Drug Discovery Foundation Scientist of the Year, Woman of the Year by the California State Senate, Science Educator of the Year by the Society for Neuroscience, and Los Angeles Magazine Woman of the Year. She's also recognized as one of the 10 best minds by U.S. News and World Report. And very importantly, for her decades of commitment to science education for inner city youth of Los Angeles, she received the Presidential Citizens Medal. So it's with great pleasure that I bring to you Dr. Roberta Diaz Brinton.
Thank you, Dr. Bernard, and to the Women of Color Committee for nominating me for this prestigious and honored seminar, Wednesday afternoon lecture. I also would like to thank the NIA and all of its program officers who work very closely with me. Dr. Wise is in the audience, and much of what I'm going to be presenting today is in his portfolio of grants to manage. So what I'm going to be talking with you about today is the journey that we have made in trying to understand why the brain develops Alzheimer's disease. And I'll start with a bit of a summary and review that age is the greatest risk factor for developing Alzheimer's disease. The other greatest risk factor for developing Alzheimer's disease is being female, being ApoE4 positive, or having a mother with Alzheimer's disease. So there's a distinctly feminine character to the risk of developing Alzheimer's disease. We know that there are multiple etiologies to late onset Alzheimer's disease, and therefore there will be multiple prodromal phenotypes and multiple progression trajectories. The field has moved on from a single mechanistic hypothesis of Alzheimer's disease to understanding that there will be multiple phenotypes and genotypes of risk, and that therefore there will have to be a portfolio of therapeutics that target these genotypes and phenotypes. The challenge for us now in this age is to develop animal models and cellular models that are predictive of the phenotypes of risk and the different stages of trajectory in the disease, and then to develop targets that we can then interrogate chemical space that hit those targets and apply that to the appropriate genotype and phenotype. The lessons that we've learned during our decades of research in this domain is that the aging brain is characterized by transitions, and in the female aging brain, transitions in the bioenergetic phenotype that involve a set of sequential system-level adaptations. The aging brain is not a linear declining brain. It is, in fact, a very dynamic brain. It is a brain with survival backup mechanisms, which it actually brings online. That female and male brains bioenergetically age differently, and that perturbing one component of this system does not create a system correction. It creates a system adaptation. And that's important when we think about the development of therapeutics. That in fact, most likely what we will have to do in Alzheimer's disease is to develop systems biology therapeutics. And lastly, there are windows of opportunity. There are transitions of aging in which there is an opportunity for a therapeutic intervention to prevent or delay Alzheimer's disease. And there will be transitions for treatment of the disease. Further, one-size therapeutic will not fit all and will not fit for all time. So those are the kind of take-home messages from my talk that I'll start with reminding everyone that precision medicine actually starts with sex. Every cell, every single cell has a sex in our bodies, including the brain. So when we look at the prevalence of Alzheimer's disease, 33% of persons with Alzheimer's disease are men, and 67% are female. And it's that female cohort of persons with Alzheimer's disease or at risk for developing Alzheimer's disease that has taught us a great deal about the disease, why it starts, how it progresses, and perhaps how we can treat it. So how did we begin this process? Well, we know that the average age of diagnosis for Alzheimer's disease is age 75. Further, we know that there's a 20-year prodromal phase to this disease. So we started to begin to look back in time to try to understand what was happening in the aging female brain that would elucidate the risk for developing this neurodegenerative disease. And it turns out that about 20 years before the age of 75, women undergo the menopausal transition. And we think about the menopausal transition from a reproductive perspective, but I will show you that the menopausal and perimenopausal transition is actually a neurological transition. Further, we began to look at the transition state that precedes the perimenopause, or the menopause, and that is the perimenopause. Because it is this transition state that we think is the harbinger of risk for developing Alzheimer's disease. So let's think a little bit about transition states. So we're all very familiar with the transition state of puberty. Puberty is an endocrine transition state related to reproductive capacity in both females and males. However, the symptoms of puberty that become problematic are not reproductive. They're neurological. An increased risk of schizophrenia in males and an increased risk of depressive illness in females. Women can also undergo a second transition, and that is pregnancy, which lasts almost a year. The last aging transition, endocrine transition, is the perimenopause to the menopause in the female. So while everyone goes through the puberty, women uniquely experience the transition states of pregnancy and perimenopause. And when we think about transition states, we think about the definition of transition states in which there are populations that are tightly controlled. And in this case, I'll be talking about brain metabolic control. And then there are populations that begin to move away from that tightly controlled system. And they begin to become more and more at risk for moving into a disease transition state. And it is this transition state that I will spend most of my time talking about today. So we know that when the brain undergoes the perimenopausal transition, there's in fact an increase in the variability of brain responses in the female aging brain. And coincident with that variability in neurological function is a decline in brain metabolism, represented here by FDG PET. So it's not a linear decline, it is in fact a step function, and that turns out to be important to consider. We know that about 80% of women who are transitioning through the perimenopause are symptomatic, 20% are not, and that of those symptoms of the perimenopause and menopause, 30% of this 80% will experience hot flushes only, and 70% of women will experience symptoms of cognitive dysfunction and cognitive dysfunction plus insomnia, mood disturbance, can have in severe cases suicidal ideation and depression. So these are all neurological symptoms associated with what we typically think is a reproductive transition. So for us, the perimenopause is in fact a neurological transition. We've spent many years understanding estrogen action in the brain, as obviously because the estrogen is an important steroid hormone related to endocrine function and reproduction in the female. And what we found over many years of research is that estrogen is a systems biology regulator of the bioenergetic system of the brain. I call estrogen the queen of Darwin. She has left nothing to chance. Estrogen is regulating glucose uptake into the brain, glucose metabolism in the brain, the generation of acetyl-CoA to generate through the TCA cycle, and ultimately to generate NADH, to generate electrons, to move down the electron transport chain, and ultimately to generate ATP. And I will come back to this whole bioenergetic system throughout this lecture. Moreover, estrogen is suppressing the ketogenic system of the brain. Estrogen is maintaining the ability of the brain to utilize glucose as its sole bioenergetic fuel. And that turns out to have significant implications. So how did we actually begin to think about the perimenopause as this vulnerable transition state? So for many years we've been conducting analyses in wild-type mice and in the triple transgenic Alzheimer's mouse. And what we see here on mitochondrial respiration is the well-recognized, reproducible decline. And I should mention, all of my experiments are conducted in females, except for the human bioinformatic analysis that I'll talk about at the end. So all of this work is generated from the female, and as in the male and the female, both show an age-related decline in mitochondrial respiration. In the triple transgenic mouse model, female, we see that even at an early age, there is a deficit in mitochondrial respiration that is maximal at 12 months of age. So for a long time, we asked, why is it that this triple transgenic mouse has such a good respiration? Because it looks just like the wild type. Why isn't it more of a linear, decline? That turned out to be the wrong question to ask. The right question is, why is it that this normal mouse looks like a triple transgenic Alzheimer's mouse in terms of its mitochondrial respiration? And it was then that we understood that, in fact, this is exactly the time when these animals undergo their mouse perimenopause and their transition to reproductive senescence. These early studies led to a very extensive analysis that we recently published in Neurobiology of Aging, in which we generated a custom targeted array around the bioenergetic, inflammatory, and redox system of the brain. And you can't read this, but essentially what I hope you can see is here in this bioenergetic component of the array, from glucose metabolism to mitochondrial function. We look here, and this is, the first line is 6-month-old animals, and then we controlled for age. We looked at animals that were 9 months of age, and they were either regular cyclers, irregular cyclers, or menopausal and no longer cycling. And I think what you can see, red is high and green is low relative gene expression. I think what you can see is that in the case of the irregular cycling females, there is a consistent and persistent decline in the bioenergetic system of the brain that then will recover, and I'll talk more about that in a moment. So there are a number of genes that reach statistical significance, and what we see again here is, in terms of the bioenergetic genes, they are primarily downregulated, and that there is a recovery of gene expression in the bioenergetic system once this perimenopausal transition has completed, and I'll talk more about that in a moment. And here what we see is a principal components analysis in which we see between these animals that are 6 months of age, and then this is the chronological age decline in bioenergetic gene or bioenergetic function. At 9 months of age, both these animals are regular cyclers, so this is not endocrine, this is chronological aging. And then here these animals are the same age, and we see a rather dramatic drop in the bioenergetic gene expression that then recovers in terms of the number of genes that are being activated here. And then at the 16 months, when these animals, again, are in chronological aging, this perimenopausal mouse age, 9-month-old animal, has a bioenergetic profile of a 16-month-old animal, suggesting that this is an accelerated aging event in the female aging brain. So in terms of whether these genes are actually predictive of changes in protein, we see that the transporter for glucose into the brain, GLUT3, is significantly reduced during this irregular cycling, as is phosphodehydrogenase, as is ATP synthase, AF2 subunit. Moreover, the insulin signaling pathway is also declined during this transition, and the phospho-CREB-ERK pathway is also decreased during this transition. So this whole system of estrogen regulation of the bioenergetic system has declined during this reproductive transition. And what's also interesting is that there was a decline in two markers of the immune system, NF-kappa B and I-kappa B. And I'll come back to the immune system. But we did see that during this time when the bioenergetic system is suppressed in the hippocampus, so is the immune system. So we asked whether this gene expression was correlated, associated with the decline in the uptake of glucose into the brain. Here you see the FDG PET signal, and we see that indeed during this transition there is a decline in glucose uptake into the brain, and the emergence of what appears to be glucose insensitivity in the periphery, glucose intolerance. Again, there's a decline in mitochondrial respiration and complex 4 activity and ATP synthase activity, and a concomitant rise in free radical production. So essentially, the brain, the bioenergetic system, this is from the hippocampus, but also in whole brain and in the periphery, there seems to be this switch from a highly efficient glucose metabolism system in the brain to an inefficient glucose metabolism system, and the emergence of insulin resistance in the periphery. So what about, these are all relevant to genes that are coded by the nuclear genome. What about the mitochondrial genome? And what we know from a number of studies is that estrogen regulates not only the nuclear genome, but also the mitochondrial genome. So let's think a moment about the mitochondrial genome. Every cell has a mitochondrial DNA derived from the mother. So its mitochondrial DNA is maternal. There are thousands of copies of mitochondrial DNA per cell. As you all know, mitochondrial DNA encodes for 13 proteins of the electron transport chain, and that the mitochondria are dependent upon about 1,000 to 2,000 of the nuclear encoded proteins that then are transported into the mitochondria. So I won't go through a lot of more about the mitochondria, except to remind people that the mitochondria are actually the starting place for steroid synthesis and fatty acid oxidation, that both of these occur in the mitochondria. Moreover, gamma secretase, that enzyme that will generate beta amyloid, insoluble beta amyloid, is enriched in the mitochondrial endoplasmic reticulum membrane. So there's lots that I want you to encode while we go along here. And so when we examined the impact of this perimenopausal transition on mitochondrial gene expression of these 13 proteins within the electron transport chain, we see again that there's a step function, a decline across these mitochondrially encoded genes associated with the irregular to regular cycling, and that these genes do not recover. And what we see is that there's a step function that in mitochondrial gene expression is sustained. So much earlier work had shown that persons with a maternal history of Alzheimer's disease actually had a substantial increased risk of developing Alzheimer's disease relative to those with a paternal history of the disease. Moreover, Lisa Mosconi's work has shown that individuals who have a maternal history of Alzheimer's disease here essentially have, relative to people with no family history, already show early in life, early in their adult years, regions of hypometabolism in the brain. And this is also true if it's relative to people with a paternal history of Alzheimer's disease, suggesting that there is a relationship between maternal inheritance and that unique genome that is inherited from the mother. So 75% of all ATP generated in the brain is utilized to reset the membrane potential. So why is that important? Well, if you don't reset the membrane potential, essentially move sodium ions and potassium ions against their concentration gradient, which is an ATP-dependent process, you're not able to reset the membrane potential, which means that you're not able to, again, fire an action potential. So it takes more action potentials to generate a synaptic transmission. So decline in ATP generation in the brain will be evident in synaptic transmission, particularly in brain regions where there's high synaptic activity, like the areas of the brain that you're using right now, the hippocampus in particular. So it's no wonder that the first symptoms of Alzheimer's disease are in fact an inability to encode new information, which is a hippocampal function, and therefore there's an inability to remember what has not been encoded. So with Enrique Candedas and Haz Sanjetti, we conducted analyses of the impact of this transition on the LTP, long-term potentiation. And what you see here on the input-output relationship is that, in fact, there's no difference in the input-output capacity of either regular or irregular cyclers. The difference comes in not whether they can reach maximum, which they do, is whether they can sustain maximum, and they cannot. And that turns out to have a significant impact on the ability of the brain to maintain high synaptic transmission. So to summarize this part of my talk, estrogen promotes the glucose metabolism system of the brain from uptake to metabolism to ultimately ATP generation. Where the evidence thus far indicates that this subsequent decline in glucose metabolism in the brain leads to an adaptive response. It leads to a response that is consistent with the brain starving, and that is now the dependency on the utilization of ketone bodies as an alternative fuel. So now the brain is a dual fuel system of both glucose and ketone bodies. Ultimately, the brain will essentially utilize all peripheral stores of lipids to generate ketone bodies. The brain, however, is under the ability to now utilize ketone bodies. And remember that estrogen was suppressing this system and is no longer suppressed. And our hypothesis that we generated several years ago, well, actually about a decade ago, is that the brain will ultimately utilize its own source of lipids, its white matter, as a source of ketone bodies to sustain brain function. And to remind you, again, that estrogen is suppressing this system of utilization of ketone bodies in the brain, and that the loss of estrogen essentially lifts this suppression of this ketogenic system in brain. So with Lauren Klaczynski, a graduate student in the lab, conducted her doctoral thesis to test this hypothesis that, in fact, this increase in H2O2 free radical production actually leads to the activation in astrocytes of the cytoplasmic phospholipase A2, which then activates arachidonic acid, which then activates sphingomyelinase, which then leads to the catabolism of white matter for fuel that then is transported as fatty acids into astrocytes and then generated by beta oxidation to generate ketone bodies to feed starving neurons. So what's the evidence in support of that hypothesis? So first of all, we replicated the decline in mitochondrial respiration and the rise in hydrogen peroxide from these now inefficient mitochondria, and the activation of phospholipase A2 in the brain. And that this activated phospholipase A2 is actually occurring here in astrocytes, as shown, astrocytes labeled for GFAP, in red for phospholipase A2, and the co-localization of the GFAP and phospholipase A2 in these astrocytes. And that this is evident in white matter regions of the fimbria, the cingulum, and the Schaefer collateral pathway. So do we have evidence for catabolism of myelin white matter? And excuse me. Essentially, what we find is a stage-dependent catabolism that first is evident in the accumulation of ceramides here in the reproductively incompetent, the perimenopausal animals, that then in the aged animals, we now see the rise in fatty acids and a change in the mitochondrial TCA panel, consistent with the utilization of fatty acids. So if that is true, if these lipids are being formed, if these ceramides and fatty acids are being formed from myelin, then there should be evidence for myelin degeneration. And that's shown here. This is an electron micrograph from Jania Trushina, all right. Oh. Okay, and Trace Christensen at Mayo Clinic. So this is a lower mag and a higher mag of myelin surrounding axons, and this is in animals that are reproductively irregular. So at this point in the kind of endocrine aging process, the myelin remains intact. Once these animals have become reproductively incompetent, and both these animals are 9 months of age, what we see is that there's an unraveling of the myelin that then ultimately appears as a loss in myelin actual density here in the aged animals. And that's shown here in terms of the number of axons affected in the collateral pathway, the Schaefer collateral pathway, the anterior commissure, and the corpus callosum all show this change in the number of axons that now show this decline in white matter integrity and white matter density. So if this myelin is being catabolized to generate lipids, we should see an accumulation of lipid droplets. And again, by electron microscopy, what we see is that there is a significant and dramatic rise in the number of lipid droplets in both these. This is in the corpus callosum, and this is in the anterior commissure, and shown here, these are lipid droplets. And again, we see that in these reproductively incompetent animals, we see a dramatic rise in the generation of lipid droplets. So if lipid droplets are being utilized and lipids, fatty acids, are being generated from these lipids, there should be evidence in the mitochondria for transport of these fatty acids and metabolism of these fatty acids. And what we see here is that the CPT1, which is fatty acid transporter, actually is greatly increased in the aged animals. The enzyme HADA, which metabolizes long-chain fatty acids, is also greatly increased. And ABAD, which is also short-chain fatty acid dehydrogenase, is also increased in these aged animals. So the ability to transport and metabolize long and medium and short-chain fatty acids in the mitochondria is evident. So if these fatty acids are being metabolized to generate ketone bodies, we should see evidence of a rise in ketone bodies in the brain, and indeed we do. Essentially, what we see here is that in the brain, in the hippocampus and cortex, what we see is in the hippocampus here, a kind of a linear rise in hippocampal ketone bodies, and in the cortex, a step increase in the amount of ketone bodies in the cortex, and a concomitant decline in the ability of the plasma to maintain ketone body concentrations. So these data are consistent with the trajectory that the brain, prior to this perimenopausal transition, is utilizing glucose as its primary fuel. The brain undergoes a starvation response to now utilize ketone bodies, and the suppression of the utilization of ketone bodies is lifted, and that ultimately there's evidence that the aging female brain will catabolize its white matter as a source of ketone bodies. And this is shown here, again, white matter integrity during this early phase, and that a loss of integrity, both of the myelin but also of the axon, is evident in these menopausal animals, and then is greatly exacerbated in the aging animals. So if this is true in the mouse, what is the evidence that this actually has relevance to the human? Oh, sorry. Forgot the immune part. So as I mentioned earlier, we saw in the the inflammation component that there were changes in the NF-kappa B system in particular during this transition, and that these were coincident. This immune system change was coincident with this rise in the utilization of and activation of the fatty acid metabolism system. So we recently conducted an RNA-seq analysis to determine the transcriptome from these perimenopausal animals, and using Illumina high RNA-seq strategy, we were able to detect around 30,000 genes, 20,000 of which were mapped, and essentially we see about 300 genes changed here. And then when we looked at where the significant number of genes were changed, it was essentially during this transition, during the irregular, from the regular cyclers to the irregular cyclers. And here what we see is, again, a little over 200 genes being changed during this irregular cycler, this perimenopausal transition, and that essentially it is during this transition that most of the genetic changes are happening relative to when these animals are now transitioning to being acyclic. So what was surprising to us is the activation of the immune system. We had not anticipated this. Essentially what the gene expression data suggests is that during chronological aging, there is a signal that emerges from the brain to then attract T lymphocytes to now invade the brain. And this chemotaxis, essentially, this chemotaxis system is activated in chronological aging. And then during the reproductive aging, essentially what we see is the upregulation and activation of antigen presentation, coincident with the utilization of fatty acids in the brain, metabolism of fatty acids. And then ultimately, there's a downregulation of the T cell proliferation response that occurs early in this process, and that essentially during the aging process, we see that there is T cell activation and differentiation in the brain. Right? This is in the hippocampus. So these T lymphocytes, we don't know what they're doing, but they've clearly invaded the brain to do something. And that is an area of active investigation by Manil Desai and R.T. Mishra in the lab. Okay, so now, what does this mean? Is there any evidence that this actually happens in the human female aging brain? And this is work that we conducted with Lisa Mosconi at NYU, in which she did a retrospective analysis. She went back into her data set of women in which she had conducted both FDG PET and beta amyloid plaque imaging with the PIB compound. And we had about 16 women in each group. So we had women of perimenopausal age without symptoms, women of perimenopausal age with symptoms, and then menopausal age women. And here's just a couple of examples. This is a 49-year-old female, and essentially 14 years of education. She reports subjective memory deficits and mild depression. And what she already shows is mild hypometabolism relative to the same age set of women without symptoms. This woman with symptoms shows already mild hypometabolism in the medial temporal cortex. This woman is 53 years old, again reports subjective memory deficits, hot flushes, and insomnia, and she too shows mild hypometabolism in the medial temporal cortex and parietal temporal cortex. In a woman who is ApoE3/4 positive, essentially she too shows, so she reports subjective memory complaints, and she too shows mild hypometabolism in the parietal temporal, the posterior cingulate, and the medial temporal cortex of both hemispheres. So this hypometabolism is exacerbated in women who, in this particular woman, who is positive, who has one allele of the ApoE4 gene. Moreover, she is already beginning to show signs of atrophy in the hippocampus. So the early analysis of these women show that there is, again, a step function in decline in FDG PET signal in the brain that then is sustained in the menopause. So it's more like a step function, and it's not linear decline in this case. What was surprising to us was the fact that when she looked at the PIB labeling for beta amyloid plaque, she saw the reverse. So now there's a rise in PIB labeling in the brain in perimenopausal animals, perimenopausal human women, and that now appears to be increasing linearly. We have an ongoing project to study this further. But this is somewhat disconcerting that early in this aging process, there's already evidence for a modest decline in glucose metabolism and a modest rise in beta amyloid generation. So when we looked at, and here's the kind of quantitative analysis of the FDG PET signal, and again, it's modest, but the maximum that is achieved typically in Alzheimer's brain is 25%. So already in this early process of aging, that we can see that there's already a 6 to 7% decline. So when she looked at the distribution of the PIB labeling across the brain, there was a surprising rise in the putamen and orbital frontal cortex. Well, the orbital frontal cortex was not so surprising, but the putamen was somewhat surprising until we asked, well, is there something that links up the putamen with the orbital frontal cortex? And in fact, there is. It's a white matter tract, the uncinate fasciculus, connects the orbital frontal lobe to regions of the temporal lobe, and its internal capsule actually runs through the putamen, suggesting that this area of the brain is already vulnerable. The white matter in this brain, in this region, is already vulnerable in these long white matter tracts. Okay, so now that we have kind of identified the bioenergetic system as a vulnerable system in the aging brain, how can we translate this basic science and early clinical science into detecting women who are at risk, who have not yet developed kind of symptoms of Alzheimer's disease, but in fact have risks? And so with Wendy Mack and Howard Hodes, we conducted an analysis of the early versus late estrogen intervention trial, and took advantage of this cohort of convenience and to ask the question, within a population of healthy women, can we use clinically familiar, rapidly deployable bioenergetic biomarkers to detect women at risk for cognitive decline? We conducted a principal component analysis that allowed us to identify the potential variables that explained the variance in the data set, and K-means clustering to essentially allow us to link subjects who had comparable profiles into these different clusters. The clusters were not predefined. It was essentially the data that drove the clusters. We had about 14 different biomarkers of metabolism in this cohort, and with the principal components analysis, 9 of those turned out to explain about 87% of the variability. So in this population of healthy women, no one's been diagnosed with disease. What we actually find are 3 clusters of women. There's the healthy women who have healthy metabolic control. There's women who appear to be at cardiovascular risk, and a third population at risk for metabolic dysfunction. And when we did correlational analyses with their cognitive function, what we found is that on global cognition, verbal memory, and executive function, the green are the healthy population of women, the red are the women at cardiovascular risk, and the women who are at metabolic risk already show a significant decline in cognitive function, suggesting that it is this metabolic function that is driving some of this cognitive decline. Moreover, when we looked at the ethnic composition of these clusters, that's disappeared. When we looked at the ethnic composition, the healthy group was largely, not exclusively, but largely populated by Caucasian and Asian women. The women at cardiovascular risk was largely, but not exclusively, populated by women of African origin. And this population was largely, but not exclusively, populated by women of Hispanic origin, suggesting that those ethnic genotypes actually can create a phenotype of risk for cognitive decline. So how do we actually address this problem or this intervention or intervening in this process? And so we developed a estrogen receptor beta selective formulation because we'd shown for many years in preclinical studies that targeting estrogen receptor beta, first of all, estrogen receptor beta is localized to the mitochondria, whereas estrogen receptor alpha is not, and that estrogen receptor beta inhibits proliferation in the breast while promoting neurogenesis in the brain, as well as promoting mitochondrial function and multiple indicators of bioenergetic system in the brain. So we therefore addressed an unmet need in women's health, and that is for a safe and effective alternative to hormone therapy. We did this by using in silico chemistry strategies that allowed us, we developed a pharmacophore that we interrogated a database of 50,000 natural source compounds, and then reduced our search into 25,000 of those compounds, and through an in silico approach, narrowed that estrogen receptor beta set to 30 compounds, which we then tested at the bench. And that testing led to the development of our phytoserm formulation that is composed of genistein, daidzein, and S-equal, all estrogen receptor beta preferring molecules, and all on the GRAS list, generally recognized as safe. We were awarded a grant from the NIA to conduct a clinical trial, and this is the first report of data from that clinical trial. And essentially, it was interesting, although somewhat disappointing. What we find is that there's no statistically significant difference of phytoserms, either at 50 or 100 milligrams relative to placebo on hot flush, on a composite score of hot flush. But I will note that more is not better, typical of estrogens, typical of steroids. More is not better. And then when we began to look at the data that emerged from this hot flush diary, these are the data from two participants. And essentially, you see that the data on a physiological outcome, hot flushes, is highly variable. And that there are different phenotypes of responders. So here, it turns out that a score of 2 is equal to 0. What we see is that there are 2 women here who essentially are very rapid responders, that early in the clinical trial, they were rapid responders and then stayed free of hot flushes. This woman, essentially this woman here, is a late responder and appears to be moving to a hot flush-free zone. And then over here, what you see is that there are multiple women who cycle from having hot flushes here to no hot flushes and back again. So this is very dynamic data. When we looked at the we had multiple cognitive measures. Here's just reported logical memory, delayed recall. What we see is that there's a hint, but not statistically significant. So we then conducted a secondary analysis. First of all, let me say that the pharmacokinetics are consistent with a 50 milligram and 100 milligram dose of phytocerms. So there's good, reliable pharmacokinetics across all these women, and that there were multiple measures of safety, all of which were met. When we began to then do a secondary analysis on responders, what we find is that there are about 10% of women in the placebo group who will spontaneously cease having hot flushes, like 90% of women do during the 3-month trial, but that about 30% of women who were treated with the phytocerms actually had cessation of their hot flushes, suggesting that there's a subpopulation of women who are responsive to this intervention within 3 months. We can't say whether others would have responded, but within 3 months, about 30% of the women on the phytocerm, on the low-dose phytocerm, were responsive. Moreover, that correlated with a significant increase in logical memory and delayed recall. So this is very typical for all clinical trials, that there are a substantial number of people within a clinical trial who will be non-responsive, and then a subset of those people in the clinical trial will be responders. So we have moved this approach into another arena, and in this case, this is around our translational center, Cure Alzheimer's Disease, and essentially our goal in the center is to create personalized, precision, and predictive therapeutics to cure Alzheimer's disease through a wide strategy from big data, letting big data drive our systems pharmacology, structure-based drug design, and metabolomics and genomics analyses, and then developing models, computational models to test for the efficacy and failure, to predict efficacy and failure, and utilizing IPS generated neural stem cells as a strategy to screen therapeutics. So in collaboration with Nofar Geifman and Atul Boot at UCSF, utilizing a data set derived from Lon Schneider, I'll talk about it in a moment, we conducted a meta-analysis for responders in failed phase 3 clinical trials in Alzheimer's disease. And these were studies or data derived from 19 studies from the Alzheimer's Disease Consortium Study and the Alzheimer's Disease Neuroimaging Initiative. They were integrated into a single data set. This is work done by Lon Schneider, Richard Kennedy, and Gary Cutter. And the combined data set has over 6,000 individuals. There were multiple treatment arms in this data set, and that it includes persons with Alzheimer's disease and persons with mild cognitive impairment, but largely people with late-stage Alzheimer's disease. In an analysis of responders in this combined data set, Dr. Geifman was able to detect that women essentially had overall worse cognitive performance than men and comparable age distribution. So this is not a novel finding. This is actually consistent with the increased burden of pathology in the female brain and both in ApoE4 negative individuals as well as in ApoE4 positive individuals. And that's shown here. Relative to men, women have, in this case, in the ADAS-Cog, a higher score is worse performance. So when she began to determine whether there were responders anywhere in this data set, what she discovered was, in fact, there were responders, and the responders were on statins. That there were multiple phase 3 clinical trials of statins that all failed. But within this data set, there were responders, and they were male. There was, in this large data set, there was no statistically significant difference in the females. And then, looking at specific genotypes, it turns out that the male responders were actually ApoE3/4 positive, suggesting again that this group of individuals, males in this case, that genotype can influence therapeutic intervention efficacy. In the case of women, essentially there was no statistically significant difference in women treated with Lipitor in this case. Actually, let me just back up a step and say that this is simvastatin. So this is a response in these ApoE3/4 males to simvastatin, whereas there was a statistically significant effect of Lipitor in females and only in the ApoE4/4 females. And there was a 4.1 difference in the ADAS-Cog. And for a clinical trial in Alzheimer's disease to be successful, there needs to be a movement of 2 points on the ADAS-Cog. So in this case, in the ApoE4/4 positive women, Lipitor was generating a 4-point difference in the ADAS-Cog, suggesting, again, that there are sex differences not only to a class of compounds, but to specific compounds, in this case, statins, simvastatin, and Lipitor. So going forward, Dr. Geifman and Atul Bhutt have already started to investigate the electronic medical records at UCSF. We're conducting analysis from the Rush studies, the religious orders and memory and aging project with Rachel Whitmer at Kaiser Permanente, Northern California. She will be investigating the impact to see whether she can actually see the same effect in her population, in the population from the Kaiser Permanente group. We're also looking in the UK Biobank, and Julie Zissimopoulos at USC is interrogating Medicare records to ask the question of persons who are on these statins, what is the risk of developing, being diagnosed with Alzheimer's disease? And if you're diagnosed with Alzheimer's disease, at what age are you diagnosed? Suggesting, we already have evidence to suggest that we can potentially achieve that very much sought after 5-year delay. But essentially we're going from a relatively small mega data set to all the way to millions of electronic medical records in the Medicare system. So the lessons that we've learned from the 67% is that there's a decline in glucose metabolism in the brain that happens early in the aging transition, in this endocrine aging transition of the perimenopause, that leads to a reliance on alternative fuel pathway ketone bodies. And that reliance on an alternative fuel pathway sets the brain up for utilization of its own storage of lipids as a source of fuel supply. And our most recent data suggests that there's activation of both the innate and adaptive immune systems. So the lessons learned are that the transitions of the female aging brain involve a set of sequential system-level adaptations and a change in your brain and your voice. Thank you. That perturbing one component of the system induces adaptations. Okay, did we get it?
Either one.
Either one. I'll take the coffee. That perturbing one component of the system does not result in a course correction. It results in a course adaptation. And that the female aging brain is a dynamic adapting brain. It's a surviving brain, suggesting that Alzheimer's disease in this aged population may in fact be due to an activation of survival responses in the aging female brain. Therapeutics will have windows of opportunity. One type of therapeutic will not fit all for all time. So the key points is that over 60% of persons with Alzheimer's disease are women. Preventing, delaying, and curing Alzheimer's disease in women will yield a 60% achievement of the goal. It's important for us to consider the right data, sex, the right informatics, the right questions based on sex, and the correct sex-based therapeutics at the right time to create precision medicine for Alzheimer's disease starts with sex. Thank you. I do want to recognize my team, a phenomenal group of people shown here. I want to particularly recognize Fei Yin and Jia Yao, who did much of this work, Jamaica and Lauren Klinzinski, and my collaborators, and especially the National Institutes on Aging. Thank you.
So we have a few minutes for questions before we adjourn to the library, where there are cookies provided by FAES. And I see that there are people lined up, so we'll just start on the left.
Thank you very much for your interesting presentation. I've followed your work on metabolism in the brain for a long time. I worked with Dr. Duckles and Dr. Kraus down at UC Irvine and cited your work extensively in our own studies. So now I'm working on actually a project involving ketone bodies and exogenous sources of them. If the brain's dying for ketone bodies, have you looked into just giving it to them?
So that's a good question, and it's a very good point. So there are products on the market now that essentially are ketogenic. So it's been known that the Alzheimer's brain actually is a ketogenic brain. And in fact, the triple transgenic mouse is born a ketogenic brain. So it's already dependent on ketone bodies. So the challenge with feeding the brain this ketogenic fuel, it may work. The data thus far suggests that it doesn't.
MCTs don't really do much. I mean, they're sort of...
Yeah, it's very modest. And part of the problem is that the transporters of fuels are actually declining. So we know that transport of glucose into neurons is first affected, and then there is a change in transporters for both glucose and monocarboxylates at the blood-brain barrier. So essentially what I think is happening is that there's this feedback system between the brain and the periphery. So, and again, kind of hypothesis here. We know that there's an inexplicable weight loss 10 years prior to the diagnosis of Alzheimer's disease in both women and men. Well, that inexplicable weight loss essentially is associated with the utilization of all peripheral stores of fat. That seems to work, but I think that it's time limited. And whether there is, and let me just say one other thing, is that one of the things that I thought is that the white matter catabolism would happen kind of at a uniform rate across the brain. It doesn't. It happens more like an ATM. So the brain is kind of going through its ATM store of lipids, and then it goes on to the next, and then it goes on to the next. So I think this whole process of when the brain starts utilizing its own lipids, it's just like in the periphery. There are changes in transporters, fuel transporters, and enzyme systems that may make it ineffective to give peripheral either glucose or ketone bodies. It's an area that I think is critical for investigation.
We have had some success both in humans and in animals feeding exogenous ketone bodies. It's somewhat important that they be the right stereoisomers, but anyway, I can send you those references if you're interested.
And I think timing is going to be critical.
I would agree with that.
Yeah. Thank you.
Yeah, you didn't say anything about the actual space-occupying lesions that are characteristic of Alzheimer's disease, namely plaques and tangles. And I know that you're not going to be able to study all autopsy brains because the people aren't dead as of yet. But in the animal models, have you found any comparison? Or in the meta-analysis, when you might have seen that some of them refer to autopsy brains actually quantifying or characterizing the neurofibrillary plaques and tangles, which are the space-occupying lesions of Alzheimer's? Is there any relationship between that and your hypotheses about that you're putting forth?
Yeah, so I think it's a very good point, and I think it's, I'm not clear on how to go about that, but I think your point is a very important one, is can we track the generation of the burden of pathology with the change in either enzymes required for utilization of ketone bodies and or the deposition or the loss of white matter, the generation of white matter hyperintensities.
Okay, and just one brief note that in terms of people who've had a transplantation of male who've had like a female organ put in, would that put them in an advanced category because now they're having more of a X chromosome burden? In other words, if a male gets a heart transplant from a female cadaver, he's essentially getting a female organ, so he becomes a chimera. I know it's kind of a strange situation, but it's something to think about.
Well, in that case, I would suggest estrogen therapy. That's a joke.
Thank you.
Did you, since you did the MRI and all the imaging techniques, did you measure the thickness of the corpus callosum, which is a major bulk?
You know, that's actually, we published that. We actually did that first. And you know, what was very strange, and we didn't, and that's actually led to the electron microscopy, is that we saw expansion of the corpus callosum. And you think, more white matter. Right? It turns out that it wasn't more white matter. It was essentially the unraveling of the white matter. So yes, we did the measurement of the corpus callosum area.
You have the mouse model. Did you give them estrogen in the area where they might be having the problems, and does it help them to reduce this Alzheimer's disease or delay it?
That's a very good point. And so the reality is that the brain is disconnecting from estrogen. So that's part of that transition, is the disconnection of the estrogen receptor system. Interestingly, the receptors are still there. And we don't know, and so we're actually investigating this now. What is the process by which there is this disconnecting of the bioenergetic system from the estrogenic control? And moreover, in terms of utilizing or intervening with estrogen, you can see that the timing of intervention is critical. Timing, perhaps during the transition, will be beneficial, but timing much later, intervening much later, essentially that system has disconnected. So it would not be effective. So I think your point is very well made, is that when is this intervention actually appropriate and when is it inappropriate or ineffective at best?
Thank you. Good luck.
Thank you.
So please again join me in thanking Dr. Diaz Brinton and join us in the library for refreshments.